for being here. And I really would like to thank the organizer for giving me the opportunity to present the work from my lab, this very exciting conference and beautiful center in Abu Dhabi. So the talk will be about RNAs and inheritance. And in fact, the central question that we are understanding in the lab is whether our gametes, in addition to our genome, they can also transmit other type of molecules, such as histone modification, DNA methylation, RNA, and proteins that might somehow be transferred to the zygote and perhaps has some biological activity during animal development and even across multiple generations. So in my lab, we are focusing on just one type of molecule for now, is RNA. And in this talk, I will talk primarily about this class of molecules called small RNAs. So why we are interested in the epigenetic fields about these epigenetic molecules is because, in contrast to the genome that remain constant to what we live in our life, this molecule can instead change in response to what we eat, even in the course of our life, the pathogen that we encounter, or even the time that we decide to make babies. All those inputs, external inputs, can basically change the repertoire of all these molecules that can be perhaps heritable and have some effect in the next generation. And that's why in the field we are trying to understand whether this can be a potential mechanism to even transmit environmental acquired information from a previous generation. Of course, we like to understand the molecular basis of this process, and we need a suitable animal model for addressing this question. So in the lab, we are using the nematode C. elegans. Here we go. This is Caenorhabditis elegans. There are two very important reasons why we think it's quite an ideal model for studying heritability, epigenetic inheritance. So first of all, this is one of the few animals that is self-fertile hermaphrodites. So one animal produces the two gametes, and is capable of making a lot of babies. In fact, from one individual, you can have a large brood size, like 200-300 babies. And therefore, those are all kind of genetically identical. So you have a population of twins growing on a plate. And so by this way, we reduce the genetic variation in the population, and we can study whether there are other type of non-genetic inheritance that happen. And then the second very important point is that it has a very fast development. And so from one generation to another, you don't need to wait years or months, but it's actually only 3 days. And so this is quite advantageous when you want to study multiple generational phenomenon. So I told you that we are interested in small RNAs. This is because since I started to study those molecules, and since the beginning of their discovery during the RNA interference process, it was already clear that these molecules were heritable during this process through the germline. But in my lab, we are really interested in what endogenous small RNAs can do and whether they can provide some heritable information. And this is what we have been studying in these past years. And so the primarily function of small RNAs in our germlines is actually an ancestral RNA-based immune system to protect our genome against genomic parasites like viruses or transposons. And there is one specific pathway that operates in animal germline, including us. Those are called piRNAs. Those are small RNAs loaded into this Argonaute protein called PIWI. This is for PIWI interacting RNAs. And so PIWI is really the major defense system we have against those transposable elements. And not surprisingly, if you are born without PIWI, or you have mutation in the piRNAs, or whenever animals have been produced that lacks piRNAs, you obtain sterility. And this is thought to be caused by the fact that you lack this repressive mechanism of transposon. Transposons start to be upregulated, jump into the genome during germline development, and disrupt germline function and gamete formation. However, when you do this experiment in C. elegans, something very peculiar is happening. So I told you the wild-type animal, this is a brood size assay. Basically, each dots here correspond to individual progeny. I told you it's around 200-300 babies. Now, when we do a CRISPR knockout of PIWI, surprisingly, these animals shows no sign of sterility at all. They are quite fertile, almost similar to the wild type. Now, if you maintain across multiple generations the PIWI knockout, now you start to observe a gradual loss of fertility until they finally, after 12 generations, upon we introduce the mutation in the population, they finally revealed the phenotype that we are used to know about pyranase mutant. And so this is quite strange because, those are all genetically identical, knockout of PIWI. They like pyranase since the beginning, but then they acquire this phenotype after so many generations. And so we were very interested to understand what is the molecular basis of this process. So the first things that we did with my PhD student, Georgia, was to look for transposable elements because the current model at that time was that still maybe transposon are upregulated, somehow disrupt germline function. So when we measure transposable elements, we developed a RNA-seq method that we could really map more than 90% of individual retroelement. But the results was quite disappointing because a really tiny fraction of those retroelements are upregulated when we remove the pyranase. And most importantly, there was no changes between early generation and later generation. So I'm going to tell you why when we remove pyranase, we don't have this massive upregulation of transposon. And this is because how pyranase operates in C. elegans. So in fact, we have 15,000 individual mini pyranase genes. Those are genes that have an identical promoter. They transcribe a huge variety of sequences. They are loaded into the PIWI protein. And by this variety of sequence, they in fact can scan and match any invasive sequence that enter into the genome, like a new transposable element. However, the pyranase are not directly suppressing those expression, but relies on a downstream machinery, which is composed by an RNA-dependent RNA polymerases. So the pyranase recruits this RDRP, uses the template to produce a massive amount of small RNAs. Those are all antisense single strands to the target transcripts. And now these are in fact the effective molecules that really silence those transposable elements. They are loaded into downstream AGO. Some are repressing post-transcriptional. Some they even go into the nucleus, suppress transcription, and even promote heterochromatin by deposition of canine methylation. Now I'm telling you this because in fact the pyranase just initiate this silencing. And so now even if you remove the pyranase, because of this amplification signaling that is still happening, this is why we believe that in absence of pyranase, you still maintain this silencing of all these elements that invade the genome time ago. And in fact, if you now remove the nuclear AGO here, now you observe 4 times more elements that start to be upregulated into this nuclear AGO mutant. Now, surprisingly, in terms of phenotype, when you measure the fertility of this knockout, there is no sign of sterility at all. You can grow for virtually infinite generation. They will never become sterile as the PIWI knockout. And so at this point, we start to believe that there should be some other things other than transposon that might explain this transgenerational loss of fertility. And so we went back to our sequencing data. And actually, we discovered a new silencing phenomenon, which was a bit odd because you remove the silencer and now you silence something else. And this is happening on a very important family of genes. Those are the histones. Those are producing core histone proteins that assemble the nucleosome. If you don't have histone, basically you end up, in fact, being sterile. And so what we observe is that there are clusters of genes that contain histone genes in the genome. They are nicely expressed, and there are no sign of small RNAs in the wild type. But then when we look at the cross-generation, we start to observe a gradual accumulation of small RNAs that are antisense to these histone genes, which anticorrelate with the expression of their mRNAs. And so we start to think that maybe this was the key to understand this phenomenon. I can tell you why, in absence of pyranase, there is a massive production of these small RNA antisense to the histone. In fact, what we observed by different type of approaches is that when you remove the pyranase, now these downstream effectors are recruited specifically on the histone mRNAs, and they start to make a massive amount of small RNAs. And this is because the histone are the only protein coding genes in our genome that do not contain polyadenylated sequence at the 3' end. And there is actually a stem loop binding protein here that promotes the cleavage. And we found that this stem loop binding protein is hijacking now this downstream machinery in absence of the pRNAs and trigger this massive silencing phenomenon. And in fact, knowing the molecular biology, we could even show that if you now remove this activity by depleting these factors in a PIWI knockout that is almost sterile, we could in fact resurrect either the histone expression and the fertility of the animal. You can in fact even virtually remove the phenotype by concomitantly knock out those two factors, and the animal will never be sterile. So with this paradigm, basically, it was good for us to understand how not only genetic mutation, but it's really the inheritance of those other type of molecules, such as small RNAs, that might reveal a phenotype across generation. And was together with other people in the field that established really how small RNAs can provide some heritable phenotype. And that's in fact what we have been doing in the past few years to understand how, germline provides some source of heritable molecules to maintain animal fertility, embryonic development, and so on. However, in recent years, other research have highlighted how even the environments that the mother experience can also have and trigger some heritable phenotype, such as stress, infection, and nutrition, which is now rising a very important point of how small RNAs that are in the soma can somehow trigger this information through the germline and to the next generation. And so we have been wondering a lot about potential mechanism that how small RNAs can be directly delivered from the soma to the germline. And so we end up studying a process that is already known in all ovipar species. This is, in fact, the pathway that produces the yolk, which is a very important nutritional substance that is composed by one type of protein, vitellogenin, which binds lipids, and is the major nutritional source for the growing embryo. So the vitellogenin is assembled with lipids, always in a somatic organs. Fish is the liver, Drosophila is the fat body, in C. elegans is the intestine. It's secreted as a large particle of lipids and vitellogenin, and there is a specific receptor for vitellogenin which taken up this substance and deliver to the next embryo, and that's how it's inherited from the intestine. And so we start to wonder whether this carrier of lipids can also transport RNAs. And this is not kind of a new idea. In fact, in the past, like more than 50 years ago, it was already like questioning whether the yolk can contain even nucleic acids. And there was a highly debated process going on, a lot of papers saying yes, no, and this was one of the last papers in the 71, they finally, by metabolic labeling in Xenopus embryo, they observed that on these yolk aggregates on the surface, you can see some puff of RNAs. Of course, we don't know what RNA is this one, whether it comes from the embryo, whether it comes with the yolk. But based on this old data, we decided to explore whether this yolk can really be a carrier of some small RNAs. So to investigate this process, with my postdoc Metali and my PhD student, we decided to develop a strategy to just trying to purify the yolk before it's getting into the oocyte to understand whether they are carrying something from the intestine. And so we first of all labeled the worm with a vitellogenin protein labeled with GFP, so we could track the yolk. You can see that in the body cavity there is almost nothing because it's rapidly taken up by the oocyte and then is maternally deposited in the embryo here. And so we decided to temporarily inactivate the receptor of the yolk. And now you can see this animal, they are still capable of secreting the yolk. It's accumulating only in the body cavity, but it's not taken up by the oocyte and the embryo. And so we developed a strategy to purify the yolk, starting from this animal, several ultracentrifugation, those are large aggregates, And then because they are also labeled with GFP, we can FACS sort, and so we can basically sequence small RNAs. So I'm showing the three major classes of small RNAs that we try to find in this yolk. So those are the pRNAs I told you about. Those are the secondary small RNAs. None of them are enriched, and in fact, those are highly present in the germline, so it makes sense that in the purified yolk we don't find them. But there is another class of small RNAs, those are called microRNAs, that are quite heavily enriched in the yolk compared to the unsorted fraction. And so microRNA, I don't know if you know them, but those are very important molecules that regulate gene expression during animal development. And so from this experiment, we start to think that maybe there is a contribution of microRNAs, not only from the germline, which is the only known tissue that give rise microRNA to the next generation, but maybe potentially through the yolk to others, for example, from the intestinal cell. And so to prove this, we developed several experiments and strategy. I just show you one experiment where we created temporal inactivation of the receptor during development to generate embryos that either contain yolk or embryo that do not contain the yolk because we deplete this receptor. And now we collect two-cell embryo. Why two-cell embryo? This is before zygotic genome activation. So if we sequence microRNAs in this two-cell embryo, we know that we are sequencing the maternal inherited one. And so if there is no contribution of microRNAs from the soma, we should see no microRNA changes. But in fact, we do see some changes. We see a fraction of microRNAs that are depleted in the embryo that do not receive the yolk. And what is important is that those microRNA are all the ones that we found in the experiment of yolk purification, suggesting that there is a transfer of microRNA from the yolk. And another important thing is that all the germline microRNAs that we know, they are intact. So this treatment is really not affecting all the germline inherited microRNAs. So with this other experiment, we think that there is indeed a maternal inheritance of microRNAs from the intestine, which is quite interesting. And the things that I want to spend a few minutes now is to tell you why the mother is transferring those microRNA from the intestine, why the germline is not sufficient to provide all the heritable microRNAs. And so what is interesting is that the yolk is in fact sensing the environment that the mother is living in. And so if, for example, the mother is growing under several stress, this can in fact have a huge impact on this yolk inheritance. So I'm just showing you one example where we stress the mother for 24 hours with the osmotic stress, and this has quite a large impact on the inheritance of yolk. So you can see that embryos coming from mother that are exposed to this osmotic stress start to accumulate a large amount of yolk compared to a non-stressed mother. And so most importantly, when we sequence the microRNA in the embryo, we in fact see a larger increase of microRNA inheritance in this embryo. And what is very intriguing is that those microRNA are not the microRNA that we found only in the yolk normally, but those are microRNAs in blue that are known to be involved in the stress pathway. And in fact, they do have an impact in the next generation larvae in controlling and regulating stress genes. And so in fact, those embryos that come from stressed mother, the larvae that develop from them, they are also quite more resistant to a future stress. So here you see those larvae coming from controlled mother or from pre-exposed mother. They grow better. They can develop in presence of a high dose of osmotic stress in the next generation. And so we do think that this effect is provided by these yolk-provided microRNAs. In fact, we can design an experiment where we stress the mother, but concomitantly we remove the microRNA biogenesis specifically in the intestine of the mother. So these animals are now born with lack of those microRNA inheritance into cell embryo. And you see that when they develop, they fail to have this stress resilience phenotype that we observed in the control animals. And so with this experiment and others, we are now pursuing the idea that there is indeed an inheritance of microRNAs from the intestine, and this might be a system to couple a nutritional source to molecules that have very important gene regulatory function in the next generation to prepare the future progeny to an environment that the mother has lived in. And I want to point out that this is an effect that lasts for one generation, is not continuous, because in fact microRNAs do not have this capacity of amplify their signal. And I just want to leave you with a video that pointed to another potential way of delivering yolk. In fact, the mother not only transferred the yolk through the intestine directly to the germline, but after they finish to lay all the embryo, the mother lived longer, and so this old mother still produced yolk for a long time. And for a while, we didn't understand why there is this yolk production if there is no progeny to deliver. And so in this paper, they finally realized that old mother can even release the yolk through the vulva. And so this is called ventation. And you can see now a huge puff of yolk at some point. Yes, this one. And so what is interesting is that if you now co-cultivate larvae with this old mother, in fact, they can be fed with this yolk. The substance is in the intestine. And so now it's intriguing to see whether there might be a further transfer post-embryonically from different organisms of microRNA and not only yolk. And so with this, I would like to thank the member of my lab. These are all the former lab member of the first generation. Now we are in the second generation, F2. I thank you for listening, and if you have any question, I'm happy to answer.
Thank you.